Transcription and Translation by Alexa Marks and Catherine Bergen. This papermation is displaying the central dogma, which is DNA to RNA transcription and RNA to protein translation. This slide shows a double-stranded DNA structure, a double helix. RNA polymerase synthesizes a sequence of RNA nucleotides along a DNA template strand. The bases in the newly synthesized RNA strand are complementary to the DNA template strand. The RNA strand peels away from the DNA template strand as DNA strands repair and wind up. RNA polymerase reaches a termination sequence and releases completed RNA strand. In RNA, uracil replaces thymine. It releases the RNA strand and the RNA strand is free to move from the nucleus to the cytoplasm to meet up with the ribosome. The DNA molecule rewinds in a double helix. RNA polymerase may now move to another gene and begin transcription. Each gene consists of exons, multiple segments of DNA that encode for protein, but there are also introns, which are other segments that are not translated. In RNA splicing, the introns are removed, and an mRNA molecule with a continuous coding sequence is formed. This is a small subunit, and it is a binding site for mRNA. Now there is a large subunit, and this is a binding site for two tRNA molecules and the catalytic site for peptide bond formation. The P site for polypeptide holds the tRNA carrying for growing polypeptides. The A site for amino acid holds the tRNA carrying the next amino acid to be added to the chain. The large ribosomal subunit binds to the small subunit with mRNA between the two. Methionine tRNA is in the first tRNA site, the P site on the large subunit. The second tRNA enters the second tRNA site, the A site on the large subunit. Which tRNA binds depends on the ability of its anticodon to base pair with the codon in the mRNA. The tRNAs with the CAA anticodon carry an attached valine amino acid, which was added to it by enzymes in the cytoplasm. The amino acid of the first tRNA attaches to the amino acid of the second tRNA. The catalytic site forms the peptide bond between the two amino acids, and a protein is starting to form. The empty tRNA is released, and the ribosome moves down the mRNA one codon to the right. The tRNA that is attached to the two amino acids is now in the first tRNA binding site, and the second tRNA binding site is empty.